Hey, welcome to day 65 of our COVID vacation, and it's going to be ETD 55 or 65. You can see that you would have had a bunch of papers you pick up for today. We'd be finishing off GR 42. You would have done GR 44 for today, although we'll come back to it. And then you're going to be picking up GR 43, the immune system, one of the most complex, but has a lot to do with the coronavirus and how our bodies are trying to fight it. We would have gotten ready for the respiratory lab that you're going to be doing in class or would have done in class today, and as well as the FRQs. You can see that the next review session will be session number 3. That'll be on Tuesday the 28th when we go after the 2014 FRQ, it looks like. Might have messed up some dates, but hey, you'll get over it. Now, key things, of course, the target for this, as we usually be coming back from spring breakish, would be explaining the process of gas exchange in mammals and describing how it impacts blood pH. You can see that one of the key ingredients in this lesson would have been bad music supplied by the teacher, like Cotton Eye Joe and Shake Shake Senora and Ghostbusters, but we don't get to do the lab. So you can find your own bad music and do the lab at home as well, because it makes exercising by yourself less awkward. What we'd be going after, of course, would be this giant legomorph. Terrifying. Poor Finny. Oh, poor guy. That, of course, is a whining child. This is a cute, happy, healthy baby. He's so happy because, of course, he's unlike this baby over here. So I'm going to get rid of my ugly head. This is a perfectly healthy baby, and this is such a grumpy baby. Why is that baby so grumpy? Well, because he has a congenital heart disease, which unfortunately is fairly common for humans. So if we consider that baby, you're going to tell me why congenital heart disease is so common in human infants. And then you can see I have the chart down there below. I want you to give me some of the major respiratory, circulatory, and digestive system differences between tadpoles, frogs, and humans. Frogs and humans you should have an experience with. Tadpoles you can think a lot like the fish that you dissected. So pause me. Write down the questions. I'd make the chart big, like an entire page, and see what you know. And then we'll come back together right now. So if we dig into this question first, you could sketch the tadpole, the frog, the really bad human with the buck teeth like Neil. Right there, if we look at them, and we can use those drawings as we go through this. Key things we should notice, of course, is if I'm looking at the respiratory system, that's about gas exchange. And a tadpole, of course, he has gills, just like the fish that we were looking at. But he also breathes through his skin. That's one of the reasons why when you touch a tadpole, they feel slimy and their skin's really, really fragile, so it can let oxygen diffuse through it. This makes perfect sense because they live in an aquatic environment. So they can keep their outer membrane, their skin moist, and their gills are able to do that for them. If we look at frogs, of course, also have slimy skin because they also breathe through their skin. They need their skin to breathe. That's why there's so many capillaries below the surface of their skin that allows them to do that. But we would have cut open frogs at this point, and you would have noticed that they have these lungs. So they use lungs. But when you looked at their lungs, you would have noticed that they're pretty lame. In fact, the weird thing about them is they actually touch the liver, the stomach, and the digestive organs in the abdominal cavity. They're in contact with it. The one thing they don't have that we have is they don't have a diaphragm, which seems really, really weird and makes their lungs pretty wimpy. They also have lungs that are easy to get to because compared to us, they have a reduced rib cage. Kermit's rib cage is pretty lame, doesn't cover up much of it. It gives some protection, but not as much as ours does. And this is because they use this thing called positive pressure breathing. Basically, what the frog is doing is he's got a throat pouch which pushes air into his lungs, creating positive pressure, which is like a ventilator system, shoving it in there. So this gives them, of course, the ability to get oxygen into the lungs and exchange CO2 and get rid of it. But it's not as great as, say, our lungs. And our lungs are enormous. We've got very large lungs, but besides that, it's hard to get to your lungs because you'd have to crack open this thing called your rib cage. And the base of your rib cage is a diaphragm. So we have lungs with a diaphragm. And that diaphragm, of course, is just a smooth muscle. If you hiccup, you can feel it spasm. And what it's doing is it's dropping down and increasing the volume of the internal cavity of the rib cage, the ribs holding the shape of that thing. And if you increase volume, then we know that pressure decreases from chemistry last year. So by doing that diaphragm and the reinforced rib cage, we can use this thing called negative pressure breathing. And negative pressure breathing means that the atmosphere shoves into us because we create a low pressure system inside of our lungs, and we can get a lot more oxygen and gases into there and exchange gases a lot faster. It makes them more efficient, right? The other part of this we can see is the circulatory system. And when we're concerned about that, we're really caring about a couple different parts. One, the heart and the number of chambers that it has, right? And then we're worried about the number of circuits inside of its circulatory system. And you didn't do a tadpole, but a tadpole has gills because it's aquatic. It lives life like a fish, basically. It's almost pretty much a fish. So like a fish, it has a fish-like heart. It has a two-chambered heart that has an atrium and a ventricle, right? Just like we do. It's part of we evolved from them. But those two chambers, of course, are linked to just one circuit. And the circuit has two stations. The ventricle is going to push it out to the gills and to the skin, because that's going to give them the ability to pick up oxygen, get rid of CO2. In that one loop then, of course, now the blood is slowed down. It goes to the rest of the system, dropping off blood at the major organs, dropping off the muscles, the brain, all of that stuff. And it's not getting the highest flow of this because it's slowed down as it passes through the gill capillaries in the skin. But it still supplies it, right? As it drops off the oxygen to the cells and picks up CO2, it can then, of course, make its way slowly back to the atria, drop in the ventricle, and get back to the gills to start it again. It circulates the blood, but it doesn't do it in the most efficient manner, right? If we look at the frog, we got an evolutionary leap. In its life cycle, it actually goes from being a fish-like creature to an amphibian-like creature. And that means that it has two atria and one ventricle. It has a three-chambered heart. It's got the classic Valentine's Day heart, and along with that, it's almost got two separate circuits. Now, I say almost because they're still connected because they have a shared ventricle. Now, that's a problem because that means that part of it's going to go to the lungs, and part of it's going to go to the skin. It's called a pulmocutaneous circuit because it's picking up oxygen there, dropping off CO2. And when it comes back to the other atria from the ventricle, it's going to get mixed together with oxygen-poor blood in the ventricle, and then that's going to go back out to the system again. So it's nice because it definitely creates a flow and allows it to pick it up and drop it off more efficiently, but it still dopes it down with deoxygenated blood. So we've got like two-ish loops, so to speak, that are connected at the ventricle. This, of course, allows them to deliver blood a bit more efficiently, and that's why a frog can move a lot faster than a tadpole and has bigger muscles that has to fuel. We, on the other hand, are another evolutionary leap. As it freaks out, we've got two fish hearts, and that means that we have two separate, completely separate atria and two separate ventricles. And from that, we've been able to set up two separate systems. And those two different circuits means that we've got two loops to work for us and four chambers to our heart. And the beauty of that is part of our heart can go off to our lungs and pick up oxygen, drop off CO2. And the other part picks that up and doesn't dope it down and then can push it out to the system from our left ventricle. Now, the beauty of this is that means that you're flowing blood that has the nutrients that you need to the right place, increasing your efficiency. And that's why, of course, you can outrun a frog if you really wanted to, because you can supply a lot more oxygen to your cells because of this more efficient system. It's like you got two fishes to make you more efficient, right? If we look at digestive systems, you didn't dissect a frog, but if you did, you notice a tadpole, or the tadpole basically has this giant belly because what it is it's a swimming stomach, it's a swimming intestine. And if you look through their belly, it's actually transparent, and you will see this huge coil of an enormous digestive tract and this tiny little fish-like creature. They're basically a swimming stomach. And what they have is they have an enlarged cecum. So their digestive tract is really, really long, and the enlarged cecum tells you an important thing. The enlarged cecum, which comes off the base of the large intestine, is loaded with bacteria. And by having an enlarged one, like all creatures that have enlarged ones, they can hold more bacteria. And that bacteria allows them to do something that we can't do. It allows them to digest cellulose because the bacteria have the enzymes to do that. And that makes sense because this is basically a little swimming cow. This is an herbivore. That's why they have to have this enlarged system, if I can spell it right. There we go. This large digestive tract, so they have more time for the bacteria to work on cellulose, and they can eat algae all day. But then during its life cycle, it goes through a massive metamorphosis and shows you how it goes from being an aquatic organism and becomes a semi-aquatic organism. Frogs don't have the gills because they spend part of their life out of the water, being amphibious, both both lives is what it means. And being semi-aquatic, that means they can breathe through skin if it's moist, and they need to keep it moist, but they also have lungs that allow them to spend more time out of the water so they can eat what they love. And if you look at their teeth in a picture, or you look at their tiny little short digestive tract, because their digestive tract is only about as long as their entire body, which is tiny. Having a short one tells you an important thing. They are a carnivore. And carnivores don't need as long a digestive tract because proteins are relatively easier to digest. They're easier to digest than, say, cellulose, right? They also have an enormous liver and a big gallbladder because they have to produce tons of enzymes to break down all those complex proteins, and that allows them to do that. Also, the gallbladder and the bile allows them to break down the fats. So their body's adapted for their lifestyle. Go figure, right? Now, the human's a different story. Your digestive tract is not long and it's not short. It's kind of a tweener. It's 26 feet long, but relative to our body, that means it's about a medium intestinal tract. And that makes sense because one thing about us is, what do we eat? We eat both. We're omnivores. And being an omnivore, we need to have the ability to get some plant matter and have time to digest it, as well as have a shorter tract because we can also eat meat and things like that. That's why our liver is a decent size and we make lots of enzymes with our pancreas. One thing we can notice about this, of course, is we have to have a diaphragm because we can't breathe through our skin. We are completely terrestrial. Now, being a terrestrial creature, we have to have a moist membrane, that's the lungs, and that increases the surface area by having huge lungs to do that. Now, the advantages of having this huge lung capacity and the most efficient blood system with 2 separate loops and 4 chambers is we need that because we are endothermic. Being an endothermic creature, that means that we're fueling ourselves. We're creating heat from our food. That's running all of our, of course, chemical reactions. This increases the rate of our metabolism. It's why we're far hungrier than, say, a frog or a tadpole or a fish, because we're warm-blooded. That also comes at a price. That means that you need to increase the amount of oxygen that your body needs to supply and the glucose supply. So that's why we have to have a more efficient breathing and respiratory and circulatory system to support being an endotherm. If we look at both the tadpole and the frog, they've got less efficient systems and they can get away with it because they're ectothermic. And being ectothermic, they don't have to eat as much, they don't have to breathe as much because they have a reduced level of their metabolism. They're cold-blooded. The environment's determining their temperature and their rate of reactions. So evolving this more efficient system and more efficient respiratory system allowed mammals and birds to conquer the land. That's why we do so well there. At the same end of that, what we're really looking at here is the evolution of vertebrates, from our fish-like ancestors to amphibians to some time in between as reptiles, then to humans. And that, of course, can be shown by things like this drawing you can actually read. And a diagram like this, when you watch a frog go through metamorphosis, you see one thing: their eggs are basically fish eggs. As they turn into tadpoles, they are basically a fish breathing with gills. And then they eventually turn into a tetrapod land amphibian, where they have to evolve to have lungs and breathe more through their skin because they've lost their gills, and that reduces other things like their tails. So you can see in a frog vertebrate evolution from fish to amphibian to conquer the land. Now, that connects back into this baby because this baby evolved from a fish and an amphibian and a reptile. And that means that the chances are that a mammal like us has a congenital heart disease are pretty darn good, because the con means that with and genital is talking about genes. The genes, of course, means that this comes with your genes, so this is an inherited trait. Right? And what we're talking about is what's known as the TBX5 gene. That's the difference between this happy baby, this healthy baby, and this not so happy, unhealthy baby. This baby has a TBX5 gene that's turned on. It inherited the gene from its ancestors. It's actually from a reptile's gene. And that means that that gene is going to produce something for him. One of the things it creates is the wall that flows between our 2 ventricles. It's called the interventricular septal. And just like the septum in your nose that divides your nares, your nose, this divides the walls of that. He's got a beautiful 4-chambered heart that's massively efficient and can feed his endothermic lifestyle. So he's a happy, healthy baby, like most babies hopefully would be, right? But every once in a while, people unfortunately inherit a gene that is a TBX5 gene that's turned off. They have the gene, but it's not turned on. And that means a crucial thing. The TBX5 gene is a gene like any others. and genes code for proteins. So it gets turned on by a thing called a transcription factor. The transcription factor builds the initiation complex. That factor, of course, can be affected by things like inducers. That will then transcribe the gene. Transcribing the gene will turn it from DNA to mRNA. And then that mRNA will meet with a ribosome and go through translation. And when it goes through translation, it will turn into a protein, a polypeptide. And this would, of course, create the polypeptide, the protein that actually forms the septum, the dividing line. This kid's TBX5 gene was turned off, so that means that they had a heart, and it had chambers, but unfortunately it didn't make enough of the protein to actually develop the intraventricular septum. So they got a little hole that bleeds in between the two ventricles. It's called an interventricular septal defect. And what that means is this kid doesn't have a 4-chambered heart. He's got a 3.5-chambered heart. If you're looking at the heart, it should look familiar because basically a 3.5-chambered heart is a little past an amphibian. And I gave him a forked tongue because this kid was born with a reptile heart, which makes sense. Our ancestors were reptiles. So if you accidentally turn off or have something in the environment that turns off that gene, now you could actually have a gene that turned us into mammals go back to its reptile-like state. And reptiles can be a little less efficient because they have some blood flow in between that makes oxygenated and deoxygenated blood mix, doping it down. What this means for the baby is it decreases the actual blood flow of high oxygen blood. So this is a huge problem if you're an endotherm. And this kid's rest of his system is set up as an endotherm. And what that means is he has a high rate of metabolism, but he has a heart that doesn't deliver enough oxygen to do that. And what that can eventually lead to is organ failure, like your brain dies or your liver dies because you don't get enough blood flow to it. Now, what this means, of course, is that if this baby was born this way, then doctors would have to go in and with a tiny little infant with their tiny little body, they would have to crack open the rib cage. And with very delicate fingers, a neonatal cardiac surgeon would come in, take the little kid's heart, cut it open, and then sew together the wall, and then sew the kid back up, and then hope the kid doesn't die. And lots of kids do die even after a really good surgeon works on them. But there's a huge advantage now because now doctors are starting to understand that this is related to our evolution. We evolved from reptiles. So being that this is a reptile gene and we can change that, if we understand their evolution, we could actually possibly treat it better and do that with gene therapy. And how would they do that with gene therapy? Well, there's a couple different ways. One, if the kid has a malfunction TBX5 gene, you could go in with CRISPR and you could potentially rewrite and put in a functional TBX5 gene. If you want to do another step, you could actually add an enhancer that would lock on and help you build the transcriptionation complex to help turn it on. You could also take and acetylate the gene because maybe it's just methylated. It was turned off, and now you open up its holiday light, so to speak, so the gene can actually be expressed. You could also take and say we have the gene, but we need it to work. You could actually insert the actual mRNA that's needed inside of the womb because you could do an ultrasound early. They do that on every baby now if you've got healthcare insurance. And that mRNA, of course, could then turn on the ribosomes. Or you could send in some small interfering RNA that could actually block things like the inhibitors that might be causing it to be shut off in the first place, which means the kid that had this disorder, a genetic disorder, you could treat the gene problem before the kid's born. They could grow their own septum and then actually never have to go through open heart surgery, which would be pretty amazing. Now, it opens huge opportunities and gives us a chance to think about the central dogma of molecular biology, genetics, right? From gene to protein. If we look at this, this is a version that you can actually read, which is handy. You can always pause and look back at it. This is a nice diagram showing basically as we go through this, as fish evolved into frogs, they got more chambers, greater efficiency, and lungs. Then the amphibians evolved into reptiles with a 3.5-chambered lizard boy heart. And then some of those reptiles, our ancestors, evolved to have a complete septal wall with 4 separate chambers and 2 separate circuits, which allowed birds and mammals, 2 things descended from reptiles, to have a much more efficient system. Right? Now, One thing we can notice that's also an issue is the fact that we have lots of valve genetic disorders. That's the other part of it. If you have doors that leak, like part of my family does. We would have been going into GR42 in the second part of it, and the second part of it really digs into the heart rate lab, which we talked about in the last ETD that I videoed for, so I won't do it again. The second part of that is GR42, and the second part through that is pages 10 through 4, and that 14, and that's really looking at how the circulatory and the respiratory systems are connected. So what we do in class is we look at a video of the cardiac cycle, which you can look up on YouTube. But the basic parts of it is if we look at the heart and these things called EKGs, electrocardiograms, you can basically figure out what's going on with the heart. That's why doctors love them so much. Now, we've got our classic 4-chambered heart. You've got your 2 atria. You've got your 2 ventricles. And in order for this muscle to fire, it needs an electrochemical signal. And that comes first from this thing called the SA node, the synarterial node. Now, when that happens with the EKG, which has sensors all over your chest area, that's going to have a little blip on the screen because an electrical potential, a charge is going through that. That causes the atria to contract, which is great because that's going to push blood from the atria to the ventricle. There's a delay then because there's no actual connecting nerves between these two things. It's just going through the muscle tissue itself before anything else happens. And that's great because that gives time for the blood to move from the atria to the ventricle. That delay shows up on the EKG, the electrical cardiogram. It's a K because it's German for cardio, but for us it's an ECG, right? That, of course, then gives a delay, and then there's another blip because what happens next is there's another node in here that's called the AV node, the atrioventricular node that meets right where the atria and the ventricle is, and that connects into, of course, this bundle branch that comes down to Purkinje fibers, and those go into the ventricle, and that's a much bigger shot because you got a lot more nerves firing, and that causes the ventricle to contract. As the ventricle contracts, you get this spike, and then you get a dip as the nerves and muscles are recharging, and then it comes back up, and it will continue on that. If you ever look at an ambulance, you can look for this symbol. It's showing the EKG, and really what you're seeing is this little guy right here, the synarterial node. That is the pacemaker. It's the natural pacemaker for you. Some folks have a problem with their SA node where it's not firing correctly because of, say, different hormone levels or different electrolyte levels in their body. So doctors have to install an external pacemaker or an internal one. That pacemaker, the internal part of it, now actually sends an electrical charge to cause the SA node to fire, and that triggers the next response because it's not so much your nervous system telling it to do it, the heart is beating on its own. And that's why you can take the heart, pull it out of somebody's chest like Indiana Jones style, and it will pump for a while because it's actually controlling itself. Most of the heart's actual rate is controlled by things like hormones, which we'll look at soon. So that's basics of how you read an EKG, showing you there's the atria contracting, there's the ventricle contracting, and you got a delay so that there's time for that blood to flow from one chamber to the next, and another delay so that you can refill the atria and get the muscle ready. Now, other things that we can see is you normally would have your heart racing a bit faster because you'd be turning in ETDs 46 through 60, but we don't have to worry about it right now. ETDs are for you right now, so hopefully you're doing your scars too. Instead, we do a lab that's one of the funnest of the year, where you would pant like a dog and you would dance around and exercise to see how that affects, of course, the pH of your blood. And this is the respiratory carbonic acid lab. It's on pages 134 through 5. And since you're probably not going to do this lab because you don't have phenolphthalein at home, here's understanding the basics because they might throw it at you. In one part of the experiment, you would pant like a dog, like this little pug, and you go breathing really, really fast, hyperventilating essentially. And you'd start to feel your body feel differently. You can try. Hopefully you don't make yourself faint, do it sitting down. The other part of it is you would exercise for a good 5 minutes with some really bad music going. And as you do that, you would start to notice you'd start breathing heavier. And you'd start to, if you're really working out, feel that burn. And in both situations, your blood starts to feel different because you're changing the pH of your blood. So you can try the lab just with the feeling parts of it, not the phenolphthalein. But I'm going to break it down for you. Basically, you would have beakers, and the phenolphthalein is an indicator of the pH. It turns pink if it has a high pH, if it's a basic solution, right? Not a lot of hydrogen ions, not a lot of protons. So if we look at that, your beakers would have started off pink as you added that stuff to it. If it goes clear, that means there's a low pH. And what we're doing is we're adding sodium hydroxide, a base, to take any acid that's in there and gobble it up in proton football, to gobble up those hydrogens and receive them to bring it back to the pink that it started at. So the data that we would see in the lab, just to give you an idea that goes into this idea, if we look at CO2, which is what we care about in this, it's going into your blood, which would have been the water in your beakers, right? Or in your own blood as you feel it. Now, CO2 is a gas, and the gas solubility is a little bit different. It becomes this stuff called carbonic acid when it goes into aqueous solution. That's dissolved CO2, just like in just like in chemistry class when we do the Alka-Seltzer lab. Now, being an acid, it's a proton donor, quarterback, right? And what it does is it dissociates, and it creates hydrogen ions, protons, and it creates these things called bicarbonate ions, which are an awesome chemical we'll talk about soon. The key thing of this is, as that's doing it, as you build up CO2 in your blood, that would cause the blood to have more protons because it would make more carbonic acid. That makes the pH go down, which means the blood is acidic. Lower pH, more acidic, more protons, right? In the lab, you would do some basic things. You would have a control that you would then have and add drops to it till it turned just pink. That would take about 9 drops standard. That's a one that you can control and compare to the others to see what your pH would be like before you change your behavior. In the first experiment, you pant like a dog, right? You hyperventilate, basically. And when you do that, there's a key thing. You're breathing in and out really, really fast. And what you're doing is you're pushing CO2 out of your body if you breathe a lot. As you do that, your blood will feel different because you're decreasing the amount of CO2 you're putting into the environment. So by losing CO2, that means that your blood is now going to make more CO2 because of Le Chatelier's principle. That's going to make you less acidic because you have less CO2 into your body. That makes your pH go up. That's why you feel funny and you become more basic. So that means that your blood is basic, and that's why it feels weird when you hyperventilate like that. That's called alkalosis. You're becoming alkaline, basic, and that's a problem because if it's too basic, that can denature proteins and make you faint because you got a problem. Your body is not working correctly. You're not going to be able to use all your receptors and things like that. That means that when we do this, the increased breathing reduces the amount of CO2 in your body, making your blood less acidic. And if it's less acidic, that means you need less base to neutralize it in the beaker, which means you need less than the control to bring it to, because it actually had less CO2 than just what was in the atmosphere or in your body to start off with. The other part that's the most fun is you'd be dancing at Cotton Eye Joe, and when you do the exercise part of this, it almost says the word exercise, the key thing is you're working, right? And if you're dancing and working, that means that you're going to need energy, so you got to do cellular respiration. By exercising, you increase your cellular respiration. And that means you're burning sugar, which means you're going to make more CO2. More CO2 turns into more carbonic acid, so that means you drop your pH and you become more acidic. You feel the burn because you're literally burning because your blood is becoming acidic. So that means, of course, that's acidosis. This is why the best runners are the best breathers. They need to get efficient in transferring CO2 out of their body, and that will help to get rid of the burn as you literally get rid of it from your body and reduce the amount of carbonic acid in your blood. So basic idea of exercising increases cellular respiration, which makes your blood increase in its amount of CO2. More CO2 means more carbonic acid, and you're more acidic, so that means you would need more drops of base to neutralize it. And it works pretty reliably. It would take about 15 depending on how much you exercise. So notice, of course, the ability of your body to get rid of CO2 has a huge impact on the pH of your blood. And if the pH drops too far, then all the proteins that make all your receptors and all your enzymes work have a problem because now you're going to cook them like bacon. You're going to denature them in acid. That's why your body is always trying to reach an equilibrium between these two things. And that's why when you run really fast, you breathe harder. Because what you're doing is you're trying to get the CO2 out, and your body says, "Hey, stop running!" Oh, and you stop, you grab your knees, and you breathe heavy because now you're losing less cellular respiration, so you make less CO2, and you're breathing CO2 out, which helps to take that carbonic acid and break it so that the universe can make more CO2 and you can get rid of it. Le Chatelier's principle, where you take and it moves towards, right? Good old chemistry. Now, the other side of this is this idea of alkalosis. And when somebody's hyperventilating, what people tend to do is give them a paper bag. And it actually does work because, one, it slows down your breathing. Usually it's because of anxiety, maybe an AP exam's coming up or something like that, or Mr. Wright just gave a whole bunch of calculus homework. But the bag basically traps the CO2 in, so you take it and it forces the CO2 back into your body. You are hyperventilating, making yourself too basic by losing CO2. It pushes more back in, which helps to neutralize you and calm you down. Now, if you don't stop hyperventilating, you become too basic and then you faint. And then the cool thing is your body kicks into automatic mode where it slows your breathing rate because it's like, hey, you're not doing a very good job, idiot. You're going to kill us. And then your brainstem takes over and slows down your breathing rate. So then you neutralize yourself out and you regain consciousness because now you're not basically dropping your brain into a base bath, right? Alkalosis. It's almost like our body understands things better than us because it's been evolving for billions of years. Now, good news. Today was about the idea of how respiratory systems affect things like our blood pH. We'll dig more into that next time with greater detail. And also this was about the evolution of vertebrates and understanding how the different number of chambers in their heart or circuits determine whether or not they're an endo or an ectotherm, how looking at their digestive system in terms of length can tell you if it's long, they're an herbivore, if it's short, then they're a carnivore, and if it's medium, they're an omnivore, and they can take over the land and try to eat everything while they do that. Hey, humans. So hopefully that helps out. You can, of course, reference the PowerPoint that goes with this for notes that are much clearer. Our next review session will be number 3. It's on the 28th, and we'll be going after the one that'll be assigned on the day, the 2014. But before that time, you got the 2013, and you're going to start working on the immune system, which is one of my favorites. Hope that was helpful.